and welcome to Spotlight on Antibodies, a podcast from BioWingtron, where we bring you the latest antibody industry trends. I'm your host, Jovina Celestio, and this month in December, we'll highlight the antibody startup ecosystem, microfluidic strategies driving next-generation antibody discovery, and the landscape of psoriasis treatment. Over the past several months, venture funding activity in antibody-focused biotechnology has remained robust, with companies securing capital from seed through late-stage Series D rounds. What stands out is not only the scale of investment, but also its breadth. Today's antibody startups are expanding from oncology and autoimmune disease to cardiovascular, metabolic, allergic, and inflammatory conditions, reflecting both growing clinical ambition and increasing technological maturity across the field. One trend driving this expansion is modality diversification. While monoclonal antibodies remain foundational, next-generation formats such as bispecific antibodies, antibody-drug conjugates, radioisotope conjugates, and engineered protein scaffolds are now central to many pipelines. Parallel to this shift is the rapid rise of artificial intelligence-enabled discovery platforms, which are reshaping how antibodies are designed, optimized, and conjugated. A significant share of recent funding has flowed into companies integrating AI at the core of their discovery engines. Chai Discovery, which raised a $70 million Series A, introduced a fully open-source de novo antibody design platform powered by large foundational models trained on diverse biomolecular structures. This approach reflects a broader movement towards computational-first antibody engineering, where structure prediction and sequence generation happen upstream of experimental screening. Similarly, Ridge Bio, backed by a $25 million seed round, is applying machine learning to advance next-generation antibody conjugates. By training models on cell-free datasets, the company aims to optimize site-specific conjugation and payload activation, addressing long-standing challenges in stability and efficacy. AI-driven discovery is also enabling more precise target selection. Immuto Scientific raised $8 million to advance a structural surface omics platform designed to identify targetable surface protein conformations. This approach recently led to a discovery partnership with Daiichi Sankyo, highlighting how startup innovation is increasingly intersecting with established pharmaceutical pipelines. Another notable player, Alchemup Therapeutics, has now raised a cumulative $114 million to apply AI toward identifying protective autoantibodies from resilient patient populations. Its clinical programs span immunology, oncology, and neurology, reinforcing the growing role of patient-derived immune intelligence in therapeutic discovery. While AI platforms are reshaping early discovery, bispecific antibodies continue to dominate clinical stage development, particularly in oncology and immune-mediated disease. Olin Biosciences is advancing bispecific candidates targeting VEGF and Ang2 for ophthalmic diseases, as well as TSH receptor and IGF-1 receptor for autoimmune eye conditions. Meanwhile, Minghui Pharmaceutical secured over $130 million in pre-IPO funding to support bispecific antibodies and ADCs already in Phase 2 and Phase 3 trials. Bispecific innovation is extending beyond oncology into allergy and autoimmunity. Dualitas Therapeutics is developing co-targeting antibodies through its platform, while Mabilon AG is advancing multispecific antibodies, including a trispecific candidate designed to neutralize peanut allergens. Besides bispecifics, IsoBio Inc. is developing antibody isotope conjugates, using therapeutic radionuclides such as lutetium-177 and actinium-225. At the same time, Odyssey Therapeutics, which raised over $200 million in a Series D, is advancing engineered V body formats and bispecific cytokine antagonists aimed at upstream inflammatory drivers. We are also seeing a rise in stealth mode companies and academic spinouts entering the spotlight. Neoc Bio, spun out of ABL Bio, launched with a $75 million Series A to develop bispecific ADCs using a proprietary Topo1 payload platform. These trends are not only accelerating discovery timelines, but also reshaping what antibody-based medicines can look like in the clinic. Coming up next, we turn from the funding landscape to the technologies powering discovery itself, as we explore how microfluidic strategies are driving the next generation of antibody discovery. From immune repertoire to functional lead candidates. Microfluidic technologies are now addressing many of these constraints. By enabling precise manipulation of fluids at the picoliter scale, microfluidics allows antibody discovery to operate at true single-cell resolution. Across recent studies, these systems have demonstrated clear advantages in accessing rare B-cell subsets, preserving natural antibody pairing, and integrating functional screening with sequencing and structural analysis. One of the most widely adopted approaches is droplet microfluidics. In this format, individual antibody-secreting cells are encapsulated into water and oil droplets, each functioning as an isolated reaction chamber. Millions of droplets can be generated and analyzed in parallel, enabling rapid functional screening at a scale that far exceeds conventional methods. Importantly, because antibodies are secreted and analyzed directly from single cells, native heavy and light chain pairing is preserved, an advantage over display-based systems where pairing is often disrupted. These droplets can be engineered to support a wide range of assays, including antigen binding, receptor internalization, and neutralization. Fluorescence-based sorting then allows rare, high-performing clones to be isolated efficiently. Downstream integration with PCR and next-generation sequencing links antibody sequence directly to functional performance, creating a powerful genotype-phenotype connection. Building on this concept, the AbDraw platform, described by you and colleagues, focuses specifically on plasma cells, an abundant source of high-affinity, affinity-matured antibodies that are difficult to capture using traditional methods. AbDraw combines droplet microfluidics with optimized plasma cell isolation workflows, processing up to 2 million cells in a single run. Within about 1 week, hundreds to thousands of unique antibody sequences can be recovered, expressed as full-length IgGs, and evaluated directly for function and developability. Other groups besides BioIntron are exploring alternatives that lower technical barriers. Researchers at the University of Cambridge developed a hydrogel-based approach in which individual antibody-secreting cells are encapsulated in agarose microgels. Secreted antibodies are captured locally within the gel, enabling standard flow cytometry to be used for sorting based on antigen binding. This strategy bypasses the need for specialized droplet sorting instrumentation while still allowing millions of cells to be screened. Using this method, potent neutralizing antibodies against SARS-CoV-2 were isolated from both mouse and human samples. Rabbit monoclonal antibodies, known for their high affinity and broad epitope recognition, have historically been underutilized due to challenges in isolating rabbit B cells. Recent work combining magnetic enrichment with droplet microfluidics has overcome this barrier, enabling efficient recovery of high-affinity rabbit antibodies against both soluble and membrane-bound targets. This expands the diversity of immune repertoires available for therapeutic discovery. Beyond screening, microfluidics is increasingly being integrated with specificity mapping and structural analysis. New platforms now combine microfluidics with ribosome display and single-cell RNA sequencing to map antibody-antigen binding relationships at scale, even without purified or barcoded antigens. At the structural level, microfluidic electron microscopy has reduced epitope mapping timelines from over a week to less than 2 days, using only microliter scale serum samples. Next, we turn from discovery technologies to clinical impact, as we examine how antibody therapeutics are reshaping the treatment landscape in psoriasis. Psoriasis is a chronic, immune-mediated inflammatory skin disease affecting more than 100 million people worldwide. Over the past 2 decades, therapeutic strategies have evolved dramatically, from broad immunosuppressants to highly targeted biologics designed to interrupt specific inflammatory pathways. Central to this evolution has been the development of monoclonal antibodies targeting key cytokines that drive psoriatic inflammation, particularly tumor necrosis factor alpha, interleukin-17, and interleukin-23. IL-23 plays a critical upstream role in the Th17 inflammatory axis, sustaining IL-17 production and promoting keratinocyte activation. By selectively inhibiting the P19 subunit, these antibodies suppress pathogenic inflammation while preserving IL-12-mediated protective immune functions. Clinically, IL-23 P19 inhibitors such as guselkumab, risankizumab, and toltrekizumab have demonstrated high rates of deep skin clearance, durable responses, and favorable safety profiles. This therapeutic class reached a new milestone late last month with the approval of pecancabart, or Pecondol, by China's National Medical Products Administration. Developed by Innovent Biologics, pecancabart demonstrated more than 80% PSI 90 response at week 16 in the Phase 3 CLEAR-1 trial. Notably, it features a 12-week maintenance dosing interval, the longest among IL-23 biologics currently approved, offering potential benefits for patient adherence and long-term disease control. Beyond efficacy, timing of intervention is becoming an increasingly important theme. Emerging clinical data suggest a window of opportunity early in disease progression, during which biologic therapy may induce prolonged remission, even after treatment discontinuation. One persistent issue is immunogenicity, particularly the development of anti-drug antibodies, or ADA. While many IL-23 inhibitors exhibit relatively low clinical impact from ADA formation, rates can vary across agents. Ongoing optimization of antibody humanization, Fc engineering, and dosing regimens aim to minimize immunogenicity while maintaining efficacy. Looking ahead, the psoriasis antibody landscape continues to expand. Late-stage development remains focused on IL-23 and IL-17, with additional regulatory activity underway across China, the United States, and Europe. At the same time, selective programs are exploring upstream or pathway-level interventions, including IL-12 or 23 P40 blockade, IL-17 receptor inhibition, IL-36 receptor targeting for pustular psoriasis, and T-cell surface modulation. As multiple IL-23 P19 inhibitors reach clinical validation, the therapeutic goal in psoriasis is gradually shifting from symptom control to durable remission. Achieving this will likely depend on integrating immune monitoring, biomarker-driven patient stratification, and strategic treatment sequencing. In that sense, psoriasis continues to serve as a model disease for how antibody therapeutics can move beyond suppression towards long-term immune rebalancing. And that's all for this month's antibody industry trends. To learn more insights into antibody drug news, make sure to follow BioIntron on LinkedIn or our website at www.biointron.com. Keep up to date with further episodes from Spotlight on Antibodies by following this podcast.